So welcome everybody again to a new global IMMUNOTALK. You probably have had the opportunity to join us before, but in case you have not, let me just remind you very briefly the goals of this seminar series. When Elena and I organized this, we really wanted to bring these wonderful talks to all immunologists possibly across the world in an egalitarian manner without the need of traveling. So these are some of the key motivations that we had in organizing this seminar series. And of course, we could not do this without the wonderful support of our global immuno speakers. We have Manuela Raffatellu today, and next week we will have Alexander Rudensky. And stay tuned because, as we mentioned before, we will have scheduled talks until the end of the year for sure, and we will be sharing with you our schedule very, very soon. So with this, I'd like to introduce my co-organizer, Elena Zuniga, who will introduce Manuela today. Eli.
Hi, Carly, thank you so much. Yeah, it's my pleasure to introduce to you today Dr. Manuela Raffatellu, a dear friend. So Manuela is originally from a paradise place, which is the province of Sassari in Sardinia, Italy. And I haven't been there myself, unfortunately, but Manuela have showed me pictures of her hometown, and it seems like an incredible place. So hopefully we can go together one day, Manuela. She has received her medical doctor degree from the University of Sassari, and then she performed a master thesis in microbiology in the same university. And in 2002, Manuela moved to the United States, where she performed 2 doctoral training experiences in microbial pathogenesis, one at the Texas A&M University and the other one at the University of California, Davis. So in 2008, Manuela was appointed assistant professor at the University of California, Irvine, and then was promoted as associate professor. And in 2017, we had the luck to recruit Manuela as a professor at the School of Medicine at University of California, San Diego. And I can say without any hesitation on behalf of my colleagues at UCSD and myself, that is a delight to have Manuela as a colleague. She is smart, passionate, very generous, and a pleasure to interact with. And many of us have already benefited from collaborations with Manuela and her team. So we are very grateful to have Manuela among the UCSD faculty. So Manuela has a long list of awards that reflect her tremendous scientific contributions, and I cannot name them all. She has received the Bureau Wellcome Fund for investigators in pathogenesis of infectious diseases. She is also elected member of the American Society of Clinical Investigation and American Academy of Microbiology. And as you may know, her work is to understand the complex interplays between the gut pathogens, the mucosal immunity, and the gut microbiota. And she has as a long-term goal to discover therapeutic targets for controlling infections. So she's interested in the host response to mucosal pathogens such as Salmonella, and she has tremendously contributed to understanding how the mucosal response to an infection is orchestrated and how pathogens evade that response, including the role of nutritional immunity, which I think she will talk about today, and represents a way in which the immune system sequesters trace minerals in an effort to limit infection. So I'm looking forward to learning more from Manuela today about mucosal defense and nutritional immunity. So Manuela, it's a pleasure to have you here, and thank you so much for your efforts to prepare this talk.
Thank you very much, Elina and Carla, for the invitation and for the wonderful introduction. It's really been a terrific place coming here to UCSD, also Elina, because of people like you. So thank you for being so welcoming. And so I love both of you and seeing both of you on Wednesday. And I hope to see each other again soon in person with both of you. And I think what this initiative that you guys are doing is terrific, and giving us the opportunity to share our science worldwide, for maybe especially at this time in which travel is difficult. So thank you both.
Thank you, Manuela. Thank you for doing this. And as always, we have a question for our speakers. We hope we can learn about them a little bit more. And the question for you, Manuela, today is related to your experience as an immigrant coming from Italy to US. What advice would you give to young scientists who are performing their training abroad?
So, and this is something, of course, that is very close to me, and probably the two of you as well, since we all moved. So first of all, I would like to say to the young scientists that are thinking about going abroad for travel, for their training, go for it. I found that the United States, contrary to what it might appear at the moment, is a very welcoming place, and it was really a wonderful experience. And I think, you know, it's difficult to know maybe early on. I think some of the struggle is whether you're gonna stay in your adopted country or whether you're gonna go back. And this is something that I think we all struggle, and it might be difficult at sometimes, but I think something to come to terms with is the fact that after you train abroad, it's difficult to have one place only that you call home. Your heart is always going to be split between two places, and I think you just have to come to terms with that. You know, the world is really your home, and you're going to be home. But for example, for me, I'm home in the United States, and I'm home when I go to Italy. But there is no, you know, there is no place, one place only that you're gonna call home, but you're really gonna broaden your mind. So it's really a wonderful opportunity, and just make the most out of it and take advantage of all the amazing scientists and all the amazing interactions that you're gonna have with people all over the world when you go to a place as welcoming and as diverse as the United States, for example.
Thank you, Manuela. That was very interesting. And yeah, I echo that feeling as an immigrant myself as well. Yes, thank you so much. So we cannot wait to hear your seminar. So if you wish to share your screen.
Yes.
Let's see. Yes, we can see, maybe presenter. Perfect. Thank you, Manuela.
Thank you again. So thank you, Elina and Carla, again for inviting me. It is my great pleasure to give this talk today. Due to the format of the talk, I'm not gonna be presenting unpublished data today. We have some work in progress from the laboratory, and I hope to share that soon. But today I'm gonna give you basically an overview about what my lab has done in the past 10 years in the terms of nutritional immunity, to understand the nutritional immunity and the inflamed gut. And as Elina mentioned in the introduction, my laboratory is really interested in the complex interactions between microbiome, pathogens, and gut immunity. And we really investigate these crosstalks and how pathogens can fight against the host to colonize, how the microbiome can provide colonization resistance to pathogens, and what are the components of the innate immune response that are either protective or that the pathogens exploit to colonize. And so what we know is that if we just think about the inflame, the regular and the gut in homeostatic conditions, so the healthy gut, is that so the microbiome in the healthy gut is largely comprised of obligate anaerobes like Bacteroidetes and Firmicutes. And you can see here that, for example, bacteria like proteobacteria, to which commensal E. coli, for example, belongs to, that constitute really a small fraction of the microbiota on the in the healthy gut. However, during colitis, and this could be due, for example, for colitis induced by pathogen, but also, like, for example, in ulcerative colitis, you can see here that there is actually a bloom of proteobacteria. So the facultative anaerobes, they are able to grow and colonize in the inflamed gut. And some of the questions that we're trying to understand in my laboratory is, so in the inflamed gut, when this environment changes, how do these microbes that bloom can compete with each other for a niche in the inflamed gut? And the pathogen that I've been working with, actually, since when I was a medical student in Italy, is the pathogen non-typhoidal Salmonella. These are leading causes of gastroenteritis worldwide. There is 100 million cases worldwide, talking about, you know, global problem. And in the United States, there is 1.35 million cases. And 26,000 hospitalization, 420 deaths a year, and 400 millions in medical costs. And drug-resistant Salmonella is also an emerging issue. As I said, this is a foodborne infection, and the disease manifestations is primarily inflammatory diarrhea, although bacteremia can occur in more severe cases, indicating again that there is a component of the host that is protective during infection, which prevents bacteremia from occurring in healthy individuals. And I'd like to highlight also that antibiotics is usually not recommended for the milder cases, like for the diarrhea cases, and is recommended only for patients with severe disease, because by interfering with the microbiota, it actually prolongs fecal shedding of Salmonella and might turn the patients into a super shed. So how does Salmonella trigger gut inflammation? And here I'm summarizing work that has been done by many people in the field, and just to give you a brief overview about what's so special about this pathogen. So in the gut, where there is, you know, healthy microbiota, Salmonella has to invade, uses a type 3 secretion system to invade, and another one to replicate and survive inside macrophages, and also inside other host cells. And as a result, what we and others have described many years ago is that there is an upregulation and secretion of cytokines like IL-23, which stimulate the numerous cells, numerous T cells in the gut to secrete cytokines like IL-17 and IL-22. And one arm of this response is to produce chemokines like CXC chemokines, CXCL1 and CXCL2. And this results in the recruitment of neutrophils to the site of infection. And neutrophils is a hallmark, in fact, of inflammatory diarrhea. This is how a physician distinguishes inflammatory diarrhea, for example, from secretory diarrhea that is caused by viruses or Vibrio cholera. And so neutrophils, so this inflamed response affects the gut microbiota, but as we and others have shown, Salmonella is able to evade this antimicrobial response and also to replicate and also grow to high numbers and transmit to naive hosts, as was elegantly shown, for example, by the Nissle monarch. So the host response, however, is also protective, because we know the neutrophils can kill Salmonella, and we know that some components of the host response are also protective and prevent the Salmonella dissemination. And so many years ago, what we found is, for example, is that during infection with the simian immunodeficiency viruses and rhesus macaques, so what we discovered is that the simian immunodeficiency virus led to a Th17 depletion, And this Th17 depletion then resulted in a blunted response to Salmonella, and this results in Salmonella dissemination and bacteremia. And other groups has also shown that this is responsible for microbial translocation. So based on, so this is, tells us basically that the inflammatory response to Salmonella is a double-edged sword, because on the one hand, it's protective and it helps to keep the infection localized to the gut. But on the other hand, it's also, there are components of the response that are exploited by the pathogen to grow and replicate. And so many years ago, we were looking to understand the changes in gene expression during Salmonella infection in the gut. And what we found is that there was a signature of genes that were very highly upregulated, both in rhesus macaques infected with, in ileal loops infected with Salmonella, as well as in streptomycin-treated mice that develop this inflammation, this characteristic inflammation in the cecum, as you can see here. And this response was a signature of genes and that included IL-22 and antimicrobial proteins like lipocalin-2 and the two subunits of calprotectin, S100A8 and S100A9, both in monkeys and in mice. And I had a very talented postdoc, Judith Benson, who's now an assistant professor at UIC, that investigated how IL-22, the role of IL-22 during Salmonella infection. IL-22 was shown to be protective, for example, during infection with Citrobacter rodentium or with Klebsiella pneumoniae, but what we found is that it actually was not protective during Salmonella infection, and it actually benefited the pathogen. And you can see here that what we found is that in mice that were infected, in wild-type mice that were infected with Salmonella, so these mice produce a ton of IL-22, we recover Salmonella from the fecal samples, here as non-lactose fermenting colonies. However, in IL-22 deficient mice, we found that E. coli here, these lactose fermenting colonies were predominant. So this indicated to us that IL-22 basically was balanced between Enterobacteriaceae that bloom in the inflamed gut. As I told you, they all grow. But if IL-22 is there, it favors a pathogen like Salmonella typhimurium over a commensal E. coli. And we were able to see this also when we analyzed the gut microbiota, because when we looked at the wild-type and IL-22 deficient mice, the microbiota baseline level was fairly similar. And however, when the mice were infected with Salmonella, you can see here that the composition of the microbiota dramatically changed. You can see the colors are totally different. And you can see that there was a high level of Salmonella in wild-type mice, and it was reduced in IL-22 deficient mice, and Escherichia instead was higher. So IL-22 was helping the pathogen. And so, as I mentioned, the other genes that were strikingly upregulated were those antimicrobial proteins, lipocalin-2 and calprotectin. And IL-22 induces the expression of these antimicrobial proteins, for example, in epithelial cells. And at the same time, neutrophils are also a source of these antimicrobial proteins. And what does this antimicrobial protein do? So one of their characteristics of these antimicrobial proteins is that they limit essential metal ions from microbes. Lipocalin-2, as I'm going to show you, limits the availability of iron, and calprotectin limits the availability of zinc and manganese, at least in the inflamed gut, as I'm going to show you. And this has a detrimental effect over the microbiota. And this is an attempt of the host to control pathogens, but many pathogens have evolved abilities to evade these antimicrobial responses and to colonize. And I'd just like to highlight that fecal calprotectin and to a lesser extent fecal lipocalin-2 are also used as a markers of intestinal inflammation and for patients with inflammatory bowel disease, because patients with colitis, they have very high level of these antimicrobials, but they're also expressed, for example, calprotectin during lung infection, skin infection. It's definitely a strong marker of inflammation. And so these responses together are termed nutritional immunity because they sequester essential metal ions from microbes. And so the term nutritional immunity was first used, to my knowledge, by Gene Weinberg in this review in 1975, and it was described as the host attempt to withhold iron from microbial invaders. And in fact, iron is definitely the most studied metal in this context. But in most recent years, Eric Scarr and others have broadened the concept of nutritional immunity to other metals as well, including zinc and manganese, and that are two metals that I'm going to briefly mention later in my talk. And so I told you that IL-22 helps the Salmonella to colonize the inflamed gut and outcompete related commensal. But what is the mechanism by which IL-22 does it? And as I said, IL-22 induces the expression of lipocalin-2, and here I'm showing you the structure and what was found in these elegant studies is that lipocalin-2 sequester bacterial small iron chelators termed siderophores, and if there are bacteria like commensal E. coli that rely on this small iron chelator here called enterobactin to grow, and then lipocalin-2 has a bacteriostatic activity over these microbes and prevents their growth. However, pathogens, including Salmonella, but also many others, Shigella, uropathogenic E. coli, Klebsiella, they actually produce so-called stealth siderophores that enable them to evade lipocalin-2 sequestration of these small iron chelators. And so Salmonella, by acquiring iron via salmochelin, is able to evade this response, and we show that this is actually very important for Salmonella growth and colonization of the inflamed gut. And the second antimicrobial that I mentioned is calprotectin. And so we've done this work in collaboration with Eric Scarr and Walter Chasin at Vanderbilt. And they had shown in 2008 that calprotectin can sequester both zinc and manganese. You can see here that is a heterodimer of the two subunits S100A8 and S100A9. And it's also highly abundant in the neutrophil cytosol, and we found it can be induced by IL-22 in epithelial cells. And as I mentioned earlier, this is used as a marker in IBD patients, and many people who work on infection actually found lipocalin-2, calprotectin in their analysis of RNA expression during infection. And so two talented students in my lab, Janet Liu and Vladimir Diaz-Ochoa, teamed up to understand the role of calprotectin during Salmonella infection. And what I'm going to briefly show you is that Salmonella evades calprotectin-mediated zinc and manganese sequestration by expressing a variety of zinc and manganese transporter, and this enables Salmonella to thrive in the inflamed gut and compete with the commensal microbiota. And so Janet, she found that neutrophils are a major source of calprotectin, and Salmonella, to evade calprotectin, expresses these zinc transporters, no ABC transporter, And this enables Salmonella growth while enabling Salmonella to better compete with the microbiota in the inflamed gut. And here I'm showing you some data. And so we did this competitive experiments in which we competed Salmonella typhimurium wild type, which is resistant to calprotectin, with its new mutant, which is sensitive, in wild type mice and calprotectin deficient mice, and also in wild type mice and IL-22 deficient mice. And you can see that what we found is that the wild type that is resistant was able to outcompete growth of the mutant in the cecal content in the feces by about 1,000-fold to 1. And this was true in both line of mice. You can see that the advantage of the wild type over the isogenic mutant was dramatically reduced in calprotectin deficient mice, the lack calprotectin, and in IL-22 deficient mice, where there was lower levels of calprotectin. So these results again indicate that Salmonella is able to evade the zinc sequestration by calprotectin as a means to grow in the inflamed gut. At the same time, so Vlad demonstrated that Salmonella is able to mitigate oxidative stress and thrive in the inflamed gut and evade calprotectin-dependent manganese sequestration. So in the inflamed gut, when neutrophils secrete both calprotectin and produce a reactive oxygen species, then this would be a very difficult environment for a microbe to grow. But Vlad found that Salmonella uses 3 transporters to acquire manganese. And manganese found is an essential cofactor for bacterial superoxide dismutase and catalase that enables Salmonella to grow in this environment. And what's interesting is that a phenotype for this enzyme was not found in conditions without intestinal inflammation. So it really seems that during inflammation, manganese becomes a really essential cofactor for helping Salmonella to detoxify reactive oxygen species and survive. And so here, so I show you some data. Again, we did a competition experiment with Salmonella typhimurium wild type, which is resistant to calprotectin, in competition with the manganese transporter mutants, and primarily CTABCB and MTH, and ZUP-T permease plays a minor role, but you can see here we use a different combination of the mutants, and what this indicates is that the wild type was able to outcompete the mutants, basically, and all of the mutants. So again, these manganese transporters are essential for Salmonella growth in the inflamed gut. And they're also important to help Salmonella to compete with the gut microbiota. And I'm showing an example here in which Salmonella was able to outcompete commensal E. coli only if Salmonella expressed these manganese transporters, because the mutant that lacked this manganese transporter here in blue colonized with E. coli at a similar level. So again, what I show you so far is that that Salmonella, the gut immune response to Salmonella is a double-edged sword, because it's beneficial to the host and keeps the infection localized to the gut. And we have some new work on components of the host response that are beneficial, but the host response is also beneficial to Salmonella because Salmonella is able to evade this response, and because IL-22 actually enhance Salmonella competition with commensal E. coli. And lipocalin-2 sequesters iron, but Salmonella is able to evade this host response, and calprotectin is able to sequester zinc and manganese in the inflamed gut, but Salmonella evades this host response, and together Salmonella can thrive. And put it in one slide. So this is the summary of what I just told you. So during, in the inflamed gut, there is a high production of IL-22, high expression of these antimicrobial proteins that sequester these essential metal ions, lipocalin-2 bind to the siderophore-antibiotic, calprotectin sequesters zinc and manganese, and the neutrophils that are recruited during inflammation also contribute to both. But Salmonella is able to evade this response by producing a stealth siderophore, by importing zinc and manganese, and this enables Salmonella to evade nutritional immunity in the inflamed gut. So after we understood how Salmonella is able to evade these host responses, we wonder whether there is something that we can do to fight against the Salmonella, because as I mentioned earlier in my introduction, there is antibiotic treatment are not recommended, especially for non-complicated cases, because they actually prolong fecal shedding. So what we did is that we looked for a strain that was able to compete with Salmonella. And this strain is the probiotic E. coli Nissle 1917. This strain has an interesting story because it was isolated during World War I by Dr. Alfred Nissle and during a Shigella epidemic. And Shigella, like Salmonella, causes inflammatory diarrhea, is very contagious, so it just takes 10 Shigella to infect a person. But one soldier basically didn't get Shigella, and Dr. Nissle was looking for, 100 years ago, was looking for strains that were able to compete with pathogens. And so this photo, actually, I took it at the 100-year anniversary of E. coli Nissle. So this meeting happened in Germany. This probiotic has been used for 100 years, and it's getting even more and more studied because the mechanism by which is beneficial are not completely understood. And we were actually interested in the mechanism by which Nissle can compete with Salmonella and potentially other pathogens. And so what I'm gonna show you is that the probiotic E. coli Nissle is able to compete with Salmonella for essential metal nutrient in inflamed gut. So in this environment where Enterobacteriaceae can grow and thrive, E. coli Nissle is better than Salmonella at growing. And the first study was done by my former postdoc, Elisa De Rio, and Elisa was able to show that E. coli Nissle has more iron uptake systems than Salmonella, and so the fact that it's able to evade lipocalin-2 mediated iron sequestration like pathogens do makes it as a good probiotic that is able to grow and replicate in the inflamed gut. And you can see that it's able to use a salmonellin, Yersinia bactin, Heme, and Irobactin, and here you can see that E. coli Nissle is simply able to steal iron from Salmonella and better compete with Salmonella in these conditions. And then another very talented graduate student, Martina Sassone Corse, showed that E. coli Nissle is able to produce these antimicrobial peptides that are called microcins, and with this peptide is actually able to kill pathogens more specifically without broadly affecting the microbiota, and is able to inhibit enteric pathogens and also other, not just Salmonella, but also, for example, adherent invasive E. coli. And So you can see here how these microcins, so these microcins are basically antimicrobial peptides that are covalently linked to a modified siderophores, and they are able to be internalized via the so-called Trojan horse mechanism, because Salmonella and other enterics have the receptor for the siderophores, so they internalize this molecule together with the antimicrobial peptide, so thinking that they are internalizing iron, but instead they are internalizing antimicrobial peptide that is gonna kill those pathogens. And so these microcins actually, and in particular one of the microcins, microcin M, worked quite well to limit fecal expansion of Salmonella in the inflamed gut. So these microcins are only expressed by E. coli Nissle during colitis or during iron limitation and colitis. So both conditions need to be present for the microcins to be expressed and be functional. So here I can see that we used a streptomycin treated mouse model of colitis, and we infected mice with Salmonella wild type, and we gave E. coli Nissle wild type or a mutant therapeutically either at day 2 or at day 5. And you can see here that we have 4 groups of mice that in the first day were all equally colonized. The first group is Salmonella wild type. Then the second group is also Salmonella wild type, treated with E. coli Nissle wild type. Third group in gray is Salmonella treated with an E. coli Nissle that cannot secrete microcins. And the fourth group is Salmonella that we engineered to make it resistant to microcins, treated with E. coli Nissle wild type. And you can see here that when we gave E. coli Nissle wild type that secretes microcins as a treatment, we were able to significantly reduce the Salmonella colonization of the inflamed gut, with some mice getting really low CFUs, like only 10 to the 2 in the fecal content. And what we found is that this was largely dependent on microcins, because you can see here that when we gave E. coli Nissle a microcin mutant, it wasn't able to reduce Salmonella to that level. And also the Salmonella that was engineered to be resistant to microcins was not killed as efficiently by E. coli Nissle. So the fact that bacteria use these Trojan horse mechanisms to kill other bacteria, that's what E. coli Nissle does, as I said, against Salmonella, but also against the adherent invasive E. coli of IBD patient, led us to a collaboration with Elizabeth Nolan at the MIT to try to synthesize these microcins to give them potentially to mice, and as a mean to specifically target, precisely target enteric pathogens without broadly affecting the gut microbiota that we know has so many beneficial effects. So we would like to spare the gut microbiota from our treatment. And so this work is ongoing, and I hope we'll be able to get the microcins in vivo, basically, to administer the microcins even without the probiotic to mice, and see whether they have an antimicrobial effect. At the same time, inspired by how microbes compete with each other, we also, in collaboration with Liz Nolan, we are using these Siderophore-antibiotic conjugates, in which we are repurposing antibiotics like ciprofloxacin or ampicillin. And so these antibiotics are attached now to Siderophore, and by the chemical modification done by the Nolan lab. And so the goal is to use these antibiotics again to precisely target pathogens without broadly affecting the microbiota. And I'll show you here some data for one of these compounds, the enterobactin conjugating to ciprofloxacin. And what we found is that this compound was very specific for pathogenic E. coli strain that expressed its IROD esterase, but it did not affect the non-pathogenic E. coli strain that don't express these esterase, and the E. coli can be engineered to express the esterase. And so in summary, so this is from a review article in collaboration with the Nolan Lab. So the type of tools that the Nolan Lab has developed and we can use in vivo to try to specifically target Enterobacteriaceae include those, include enterobactin or salmonella that are conjugating to beta-lactam, and they show that there is specificity, for example, for pathogens that express this receptor, the salmonella receptor IRON, or as I show you, we can use ciprofloxacin linked to enterobactin, and we can find that this is only cleaved when the bacterial enzyme is present, and this is in mostly pathogenic strains. And again, this is part of our effort to use Siderophore-antibiotic conjugates on microcins to specifically target enteric pathogens without targeting the microbiota. But I think that a deeper understanding also of a mechanism by which the microbiota are able to survive during infection might tell us whether our strategies might be more or less specific and what we can do to make them more specific again against enteric. And on this note, I would like to highlight a recent study from Wenhan Zhu and Sebastian Winter. So this is a beautiful study that was published earlier this year and showed that commensal bacteroides B theta iota omicron uses the siderophores that are produced by Salmonella during infectious colitis or E. coli Nissle in non-infectious colitis. And these enable these bacteroides to grow and replicate and survive inflammation and promote the reconstitution basically of the microbiota during colitis. So I think it would be important, for example, to see whether our strategies are whether our molecules are also being internalized by such commensals, because the receptors are different, so they might be specific, but of course we would need to investigate this further. And so last but not least, another approach that we've been using in collaboration with the Nolan Lab, and this work was done by Martina Sassoni-Corsi in collaboration with Puncha Raitana when they both were students in my lab and in Lisa's lab, so is to use siderophores to trigger an immune response and block iron acquisition by pathogens. So as I showed you earlier, our current model is that lipocalin-2 is able to block enterobactin and thereby reducing growth of commensal E. coli. But Salmonella has a stealth siderophore that enables it to acquire iron in the inflamed gut. So we thought about conjugating the siderophore. So these are small molecules and they're non-immunogenic, to a carrier protein. And by conjugating to a carrier protein, we would be able to induce an immune response. And we chose the cholera toxin B subunit because that's a strong mucosal adjuvant and we wanted to induce a strong mucosal response. So the idea is that we would get B cells in the mucosa that would produce antibodies that would bind to enterobactin and salmonella due to their common structure. And this way, we would be able to reduce the colonization of pathogens. And this worked actually quite well. And we were able to see antibody productions over time to enterobactin in the Pierce patches. You can see here, both by ELISA and by ELISpot. And these antibodies also, we found antibodies also that recognized salmonellin. And what we find is IgA that are strongly present in the prior species of immunized mice. And right now, a new very talented postdoc in my lab, Romana Garner, has been isolating those B cells clones, and we're trying to purify those antibodies for potentially using them therapeutically during salmonella infection. And what was interesting also about this study is that we were wondering whether there was a detrimental effect, whether there was a beneficial effect during salmonella infection, and whether there was a detrimental effect on the gut microbiota. So here I'm going to show you the colonization data of mice that were immunized either with our Siderophore conjugated with a cholera toxin B-subunit or the cholera toxin B-subunit. These are mock immunized mice in black. You can see here that there was a high level of colonization about day 2 and day 3. But you can see that at day 4, which is when the peak of the inflammatory response to salmonella occurs, we had a very significant difference between the mice that were mock immunized, in which we see high levels of salmonella colonization, and the mice that were immunized against enterobactin. You can see that there was a significant reduction of colonization, and some mice even had very low levels of salmonella, and these were the mice that produced the strongest antibody response. So regarding the effect on the microbiota, what we found is that at the baseline level, immunization with CTB or CTB-ANT did not broadly affect the microbiota. In fact, the composition was fairly similar between the two groups. During infection, what we have found is that in the mock immunized mice, there was a high level of salmonella as expected, and as I show you in the prior slide. What happened though in the mice that were immunized with the enterobactin? So in these mice, show you there was lower level of salmonella by bacterial count, and this was also reflected by analyzing the composition of the microbiome. But I hope that you can see that there is a population of microbe here that is highly represented in mice that were immunized and against Salmonella siderophores, and this was actually lactobacillus. So lactobacillus are interesting because many species are known not to use iron for their metabolism. And also other studies, for example, by Fiona Parry group, nicely shown that lactobacillus can thrive during colitis. And it would be interesting to understand by which mechanism. But what we were happy to see here is that our immunization strategy by reducing salmonella colonization didn't promote the expansion of microbes that might be detrimental, and instead these microbes are actually largely considered to be beneficial. So this is basically a summary of what I just showed you, that in mice that are mock immunized, Salmonella is able to evade the host response by acquiring iron via salmonellin, and is able to grow and thrive and colonize in the inflamed gut. However, if we immunize mice and these mice produce antibodies against the siderophores, enterobactin, and salmonellin, we reduce salmonella colonization, and this actually results in an expansion of lactobacillus, which are the microbes that take over the niche of the inflamed gut when salmonella is reduced, at least in this experiment. So with that, I would like to conclude by leaving a few key points. So what I hope you take away today from my talk is some concepts about nutritional immunity in the inflamed gut. So this is a conserved host response that they limit access to, of metal ions like iron, zinc, manganese, potentially others, to pathogens. These metals are essential for host and pathogen alike, and so during infection, the host tries to sequester these metals. And the cytokine IL-22 is actually a key modulator of nutritional immunity. And it does so by promoting the expression of these antimicrobial proteins, lipocalin-2 and calprotectin, that sequester essential metal ions from microbes. And pathogenic microbes, they have evolved mechanism to evade nutritional immunity. So this host response is probably effective against some commensals or against microbes that are not very pathogenic, because a pathogen must evade this response to colonize and grow and thrive. And likely for us also, some beneficial microbes have evolved means to evade nutritional immunity, thrive in the inflamed gut, and compete with each other for limited metals. And last but not least, as I show you with the little more translational work that we have been doing, is that understanding how pathogens evade nutritional immunity could lead to new therapies. And I think that now that we understand the importance of the gut microbiota and how basically the gut microbiota modulates everything from development of immunity to the gut interaction with the brain, I think it's really the time to make therapies more specific to target pathogens without broadly affecting the microbiota. And so with that, I would like to conclude by thanking my laboratory. I thank the people who did the work. And this photo is very special to me because it was taken on February 24. It was the last lab outing for who knows how long. And so we were at the bar nearby celebrating celebrating one of the students who temporarily left before coming back to the lab. So I really have to thank these incredible people that have worked with me over the years that I mentioned, the current as well as the current ones. I'd like to thank my funding sources, the NIH and Burroughs Wellcome Fund, and the numerous collaborators over the years. And here I'm just acknowledging the ones that collaborated during on these projects of nutritional immunity: Eric Scarr, Walter Chazin, Liz Nolan. I haven't told you about the work that I'm currently doing with Peter Dorstein on zinc in the inflamed gut, zinc and iron in the inflamed gut, Ferric Fang, Steve Levy, Rob Edwards, Celia Golding, and others. And thank you again, and I look forward to answer your questions on Twitter. And Elena and Carla, it's so great to see you both, and thank you for organizing this seminar series. I enjoyed very much, and really, it makes me excited on Wednesday morning.
Thank you so much, Manuela. This was a beautiful talk, and I definitely learned a lot. Yeah, thank you. I appreciate your efforts preparing the talk and also for answering questions in Twitter. Yeah.
Yeah, thank you so much, Manuela. I really appreciate you sharing your innovative way of thinking and approach when it comes to understanding this interaction between enteric pathogens and the commensals and the host. So thank you so much. I learned a lot. And so before we finish, let me remind you that if you want to ask questions, you can search for the Global IMMUNOTALK Twitter account, and you will already find a tweet that says ask questions to Dr. Manuela Raffatellu here. When you ask your questions, please remember to mention Global Immuno, okay? And as we mentioned, so Manuela will use her own Twitter account, which is @Manuela Raffatellu. So thank you so much. Thank you, Manuela. Thank you, Elena. And we'll see you next week with Dr. Alexander Rudensky.
Bye, everyone. See you next week. Bye, Manuela. Thanks again.
Bye. Thank you all.